Chapter 3, I'm going to do sections 3.1 through 3.3 as a flip presentation. So again, you should have your notes printed out and the first part filled in for class on Saturday. So in this chapter, we're going to talk about organic compounds. So we're going to talk about those different molecules that make up living organisms. Those types of organic compounds we're going to focus on are carbs, lipids, proteins, and nucleic acids. We'll also be doing a lab Saturday on all 4 of those biological molecules. So many molecules found in cells are composed of carbon, which are bonded to other carbons and atoms of other elements. Now, the good thing about, or I should say the versatile thing about carbon is that carbon can form up to 4 bonds. Because if we look back at our Lewis dot structure, we have carbon with 4 valence electrons. Now, generally, molecules that contain carbon are referred to as organic compounds. So we'll need to use that proper terminology when we say we have an organic molecule. We're not referring to something that has grown in the absence of pesticides or hormones. What we're saying is that it is a molecule that contains carbon. So by sharing electrons, carbon can form, like I said, 4 other bonds, and it can form in many different shapes. It can branch, it can be straight chains. So there are different types of ways that we can display those molecules. So we have here with the simple diagram showing the carbon with the 4 hydrogens. We can then actually look at the shape of that molecule here with an actual model. And then we have what we refer to as our space-filling model over here. So there's different ways that we can represent that particular molecule, which is methane. So a carbon skeleton is a chain of carbon atoms that can be branched or unbranched. So we can kind of look at that. I think I have some pictures here in the next slides. And when we talk about compounds with the same formula but different structural arrangements are called isomers, and I am going to show you a picture of that as well. If not, I definitely showed a picture of this in the guided reading. I gave you some isomers of sugar. So in this picture, I actually am showing multiple things. So we have right here the carbon skeletons of ethane and propane, and we can see they're fairly straight chains. Versus over here, we have some branching. So these are actually some isomers right here. We have butane, which is a straight chain, and we have isobutane, which is branched. But if you count up the number of carbons and hydrogens in each, their chemical formula is the same, which is C4H10. And if you look, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and our 4 carbons. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we still have the same molecular formula, but again, instead of it being a straight chain, this one's branched, but it has the same number of carbon, the same number of hydrogen. So those are referred to as isomers. We can also get some double bonds where carbons are going to share 2 pairs of electrons. So when we have double bonding, notice that the chemical formula is the same. However, we're going to have to identify where that double bond is. And then over here we have some ring structures where they have formed a circle with the carbons. So in this animation, and hopefully this will work at home, but basically what they're just trying to show you here is the different types of isomers, which is a little more detailed than we need for this class. But basically what we're saying is that the chemical formulas are the same. The arrangement of the atoms are different. And that's all you need to know about isomers. Now we have some key functioning groups in biological molecules. So an organic compound has unique properties that depend upon the size and shape of the molecule. And the groups of atoms, we're going to refer to those as functional groups. So a functional group affects a biological molecule function in a characteristic way. Generally what we'll find is those functional groups will either infer whether it's water-loving or water-fearing, so hydrophilic or hydrophobic, or if it will form hydrogen bonds. So it kind of influences some of the properties of those molecules. So these are the main functional groups. We're going to have a hydroxyl group, which is OH, which is going to be polar. It may form hydrogen bonds. And then we have what's called the carbonyl group, which again, notice if it has oxygen, we talked about how oxygen is electronegative and doesn't like to share very well with the other elements. So it will generally pull those electrons closer to it, which will make it polar. And here we have some more oxygen right there. So again, another polar group. Now here we have an amino group. We don't have that nasty oxygen atom, but we do have a hydrogen. And what that'll tend to do is form hydrogen bonds. And then we have the phosphate, which again has oxygen, so it's also polar and will form polar covalent bonds. And then we have the methyl group, which we'll talk a lot about methyl groups and how they tend to be nonpolar and how they kind of are used for sugars and kind of changing different things on proteins. We'll talk about kind of adding sugars and stuff to proteins. So here is an example of a molecule that has some of those functional groups on there. So we can see here we've got our methyl group, our hydroxyl group, and then our carbonyl group right there. And how these two molecules, other than that, they are identical, but it is a difference between the male and the female sex hormones, testosterone versus estrogen. So there are 4 classes of molecules important to organisms. So we've got our carbs, our proteins, lipids, and nucleic acids. And like I said, we're going to look at all those very closely in our lab. So you need to be familiar with them and what they're used for and their basic structure. So the 4 classes of biological molecules, and you'll often hear me refer to them as macromolecules because they're big, they're large in size. They're also called polymers, and they're called polymers because they're made up of identical building blocks that are strung together. Now, those identical building blocks are referred to as monomers. So monomers are linked together through a chemical reaction that is called dehydration. Okay. There are 2 names actually for this particular reaction. You can also call it condensation. And what happens here is we can see our polymers. We have a polymer here. Well, actually, this whole thing here is a short polymer, but what I was trying to say is you can see we have 3 monomers. So these little monomers that we hooked up into a short polymer. And we want to add this singleton here, or the single monomer over here. And in order to do that, because again, elements can only form so many bonds. So we're going to have to remove and break some bonds in order to get these 2 to hook up. So when we do that, we're going to actually pull out a molecule of water. And that is why it is called dehydration, because we're pulling water out of that molecule. And if you think about condensation when you run your air conditioner, or even when you have that water, when you say your glass sweats, when it's humid out, it's condensation. It's water that's forming on the surface of that cup. So forming water from that chemical reaction is also, again, that's another reason why we call it condensation reaction. So you do need to know that this is condensation or dehydration. And when we have that type of reaction, we are making a larger molecule. We can say it's anabolic. Anabolic meaning it is building up that molecule, kind of like an anabolic steroid builds up muscle. So here you can see the short polymer and how we're going to hook this up. And then the next slide. So in this slide, then we're just seeing, again, them hooked up and the water coming out here. So again, this is an anabolic reaction. The type of chemical reaction is also referred to as dehydration or condensation. So of course we need to have the opposite type of reaction, because there are times where we need to break down molecules, for instance, with digestion. So this is the opposite. So in order to break apart this bond right here, we have to add the water back into that molecule. And the word hydrolysis, hydro meaning water, and lysis means to split. So we're going to take water and split it and put it on the ends of our monomer here and our short polymer. In this slide, we're just seeing our original polymer, which we're then going to add water and split it. So here's the water. It undergoes hydrolysis. And now we have our monomer back and our short polymer. All biological reactions of this sort are mediated by what we call enzymes. And enzymes speed up the chemical reaction. Most of these chemical reactions will take place on their own. It just will take place on such a slow scale that it would not be useful for living organisms. So we're going to use an enzyme, which most enzymes are proteins, but not all. So we're going to take that enzyme and it's going to kind of speed up that reaction by causing additional stress on some of those covalent bonds. And hopefully this animation will again work at home and you can see that process occur. So a cell can make a large number of polymers from a small group of monomers. For example, proteins are only made up from 20 different amino acids, and DNA is built from just 4 different types of monomers called nucleotides. So we're going to use these to make a bunch of different molecules, and it's kind of interesting that we can create so many different living organisms just by rearranging the order of some of those monomeric units, especially with DNA. So we're going to talk about proteins, carbs, lipids, and nucleic acids in much more detail in class. I just don't feel that this is the format to do that. So at this point, we are going to focus in on learning the rest of this chapter in class next Saturday. So we'll again do the coursework and then go up into lab and then actually handle some of these biological macromolecules. Good luck studying this week, and I will see you on Saturday.